Lecture 15. We are going to discuss meiosis. The objectives that you should know by the end of this lecture. Sexual reproduction. This is an important way of reproducing for most eukaryotic organisms. Whether they can reproduce both sexually and asexually, most eukaryotic organisms have the ability to reproduce sexually. And this is going to involve the making of gametes through the process of meiosis. Those gametes will eventually come together to fertilize one another and produce a new organism. In sexual reproduction, the DNA of the offspring comes from two parents. The egg and the sperm, each coming from an individual parent. They will fuse together and yield the next generation. Sexual reproduction generates genetically diverse offspring. So the mixture of the DNA of the two parents will come together to make a unique offspring. So why sex? Why sexual reproduction? Attracting mates and producing sex cells, it's going to require a lot of energy. It's very costly to the organism. If you are coming into contact with another individual, there's the possibility of being their lunch. And so why do this? Well, sexual reproduction is going to create diversity in the genetics and give the offspring an extra chance of survival. Genetic diversity is the worth. Sexual reproduction produces genetic diversity among offspring. This is going to increase the chance that some of the offspring may survive during environmental changes. The graphs that we see here on the left and on the right, the left-hand side is showing a population of organisms that have very little genetic diversity. On the right, an organism that has lots of diversity as time goes on. And so if an ecological disaster happened, let's say a fire comes through, and we're talking about plants. If you have the population of this particular plant species is exposed to a roaring fire, and they're not able to withstand this fire. That fire is going to rip through and it's going to kill all of the trees, all of this particular plant, and then none of them will be moving on to the next generation. Whereas if you had diversity amongst the plants on the right-hand side, the fire rips through, just like it did over here, and it's going to kill a large portion of the population. But let's say there's some diversity amongst those plants. Some of them have seeds that are coated extra thick, and they can withstand the heat of the fire. They're not going to burn all the way through. Some of them can hold on to water a little bit more, and so when the fire roars through, the plants are able to survive better. And so, yes, a large population of this species could die off, but a small part may be able to survive, and then it will become more diverse as time goes on. Other examples, diseases, climate change, a new predator is introduced. Lots of changes can happen in the environment that can hurt the future of a particular species. So the next couple slides are reviews of some terminology, making sure that we all remember some of the terminology that we went over before. So chromosomes in different organisms different species will have different numbers of chromosomes. For humans, we have 46 chromosomes in our body cells, our somatic cells. Those 46 are actually 23 pairs of homologous chromosomes. So somatic cells, or the body cells of an organism, will be diploid. Diploid is meaning 2n. And so for humans, n is 23. There are 23 different types of chromosomes that our cells have. 2 times 23, so there are a pair of every chromosome in our cells. So a pair of homologous chromosomes, so 2 times 23 equals the 46 chromosomes in our body cells, in our somatic cells. So basically, all of the cells in our body are diploid, except for our gametes, which is our sperms and our eggs. Those are the only gametes that are found in a human body. Those cells are haploid. They only have one N. They only have 1 times 23. So they have half the DNA that our body cells have. And these cells, these gamete cells, will be created by meiosis. Haploid cells will be created by meiosis. So in the last lecture we talked about mitosis, creating identical cells. The 2 daughter cells from the parent cell will have the same number of DNA, and in meiosis it will not be this way. Meiosis, the parent cell, will have the 2n, but it will create 4 daughter cells that will have half the DNA of the parent cell. And we'll talk about how this happens through all the different steps of meiosis. So again, somatic cells, the diploid body cells of the organism. They're undergoing mitosis and producing identical diploid daughter cells. So the parent cell will be going through mitosis and creating 2 identical daughter cells. Germ cells are going to be the cells that are undergoing meiosis to create gametes. Germ cells also have 2n. They are diploid, and they are these special cells that are going to undergo meiosis. So the parent cell that is going to go under meiosis is going to be the one that is creating these gametes. But the parent cell itself, or the germ cell, will have the 2 times 23, or the 2n. So germ cells will be undergoing meiosis to produce variable daughter cells that are going to be haploid gametes. Diploid cells contain 2 sets of chromosomes. Most cells in a sexually reproducing organism have 2 sets of chromosomes, one from the father and one from the mother. So in every one of your cells, you have 23 pair of chromosomes. Each pair, 1 came from your mother, 1 came from your father. And cells with 2 sets of chromosomes are called diploid cells. So just reiterating these terminology here. So here we have another look at a human karyotype. We saw a similar picture in the mitosis lecture, but now we'll get into some more verbiage, some more vocabulary about what things are. So again, the human body cells, somatic cells, contain 23 homologous sets of chromosomes. As you can see in the karyotype, this pair is number 1, this pair is number 2, 3, 4, and so on. So when I say that each set of chromosomes, 1 came from your mother, 1 came from your father, in this particular set, 1 of them came from your mother, 1 of them came from your father. Same with this set. and every other set. And so this image that we're seeing here is called a karyotype. It's a photograph, an image of all of the chromosomes of an organism. This is a karyotype of the human chromosomes. It shows all the chromosomes from a diploid human cell. Humans have 23 pairs of homologous chromosomes. 22 of them are autosomes. And autosomes are special because they are found in both males and females. So no matter what sex you are, you're going to have the 1 through 22 autosomes. Notice that each autosome, they are roughly about equal size. They're going to have basically the same genetic makeup on them. The different traits that is found on one will be found on the other. The 23rd is going to be the sex chromosomes. The chromosomes denoted by the letters X and Y are going to be the sex chromosomes. In humans, an individual with 2 X chromosomes is going to be female. So this here is a female, X and X. Notice that they're both the same size. They're both large in respect to one another. And then if you have an individual with an X and a Y chromosome, that will be male, and that is shown here. And so the X is the large chromosome, and then the Y is the small chromosome. And this will create a male. Mammals are this way, XX being female, XY being male. Other organisms can be different. We can have the ZW situation. I think most birds are ZW, where the ZZ will be the male and then the ZW will be the female. Some insects will have different sets and situations. Not all organisms are male-female, denoted by sex chromosomes. Some of them become male or female by outside environmental forces. When it comes to alligators and crocodiles and some turtles, the actual temperature of the egg during certain points of development is going to be what determines if they're male or female. So there are lots of different ways of doing things out there in the animal kingdom, or all of the other eukaryotic organisms. But for humans, this is how we do it. Homologous sets of chromosomes. So we keep throwing out that term, homologous set of chromosomes. What exactly is that? Well, a homologous pair of chromosomes are going to be, similar size, similar structure. They're going to have the same types of genes on them. They might not be the exact sequence of DNA as one another. Actually, they're most likely not going to be, because that is creating diversity. The DNA sequence will not be the same on these different chromosomes, but each chromosome of the homologous pair is going to have the same genes for the same traits. And so if we look at a lot of the times when we have 2 homologous chromosomes, they're going to be, one's going to be pink and one of them is going to be blue, and it's kind of denoting that the pink is what came from your mother, the blue came from your father. But however you want to see it, one came from one parent, one came from the other. I think on the next slide it actually gets into the alleles, so I'll skip ahead. I don't want to. So homologous sets of chromosomes, again, they're not identical to one another. And you can see, this one has a gene, big A, big B, and little d. And so when you look at the comparison to the other chromosome, yes, it has a big A, so that means they both have the same allele for this gene. And these genes can be, let's say, hair color. So let's say big A is brown hair, little a is blonde hair. So they're both having the same allele for brown hair. Whereas B could be eye color. And so the big B is brown, little b is blue. And so in this case, this allele, or I'm sorry, this chromosome has the allele for brown eyes, and this chromosome has the allele for blue eyes. Now, it's still the same gene that is going to create the eye color. The difference is there are two different types of this gene. The two different types of genes, or versions of that gene, are called alleles. So alleles are versions, different versions of a particular gene. And we'll talk about this much more in the next lecture when we're talking about genetics and things like that. Notice the two different alleles for B, although the alleles for genes A and C are the same. There is no C that should actually be saying D, so they're both little d's. And so when we talk about homologous chromosomes pairing up, so this could be homologous set of chromosomes number 1. And so both of these chromosomes are chromosome number 1. They just happen to be the homologous pair that are number 1. One coming from your father, one coming from your mother. Gametes are produced by meiosis, and we will see eventually how meiosis works and what it's doing to create these gametes. Gametes, like sperm and egg cells, they're going to be haploid cells. They're going to be 1N. They're going to have half the DNA of the rest of your body cells. A haploid cell has only one set of these chromosomes. Now, you might be thinking, well, if you have the homologous chromosomes, one's coming from your mother, one's coming from your father, what do these gametes have? They're going to be a mixture of the DNA from your mother and father, and we'll show how that's going to happen when we talk about meiosis. When the gametes fuse together, when the egg and sperm fuse together, this will be fertilization, and this will create a diploid zygote. So the egg and the sperm are both haploid. They're both 1N. When we put them together to fertilize one another, the 1N plus 1N will now create a 2N cell. And that will be a zygote. That zygote will be the first cell of a new organism, genetically unique to either of the parents. And then that zygote will create eventually an embryo and a new individual, from zygote to the adult, it will be going through mitosis to create the rest of that organism. This is kind of a diagram kind of showing this, where over here that fertilization of the egg and sperm is taking place. Once that happens, the blue is showing this is diploid cells that are going through mitosis, and then when eggs and sperm are created by meiosis, and then those will be 1N until it's fertilized. So showing another example, instead of keep showing humans, we're going to jump into a situation of frogs, amphibians, and kind of how this meiosis cycle is happening. And so here we have that fertilization of egg and sperm took place. And so we've got the zygote. This is the first 2N cell that is going to be this new individual. It's going to go through mitosis to create the tadpole, go through mitosis continuing as the legs grow, as the tail falls off through apoptosis, and then eventually you'll have a male or a female, whatever the genetics are of this particular individual. And so mitosis all the way up to the individual, up to the adult. So all of the cells in these adults will be 2N. Inside these adults, there will be gametes being formed. And so that is the only place where meiosis is taking place. So this female, meiosis will be creating eggs inside of her, one little spot in the ovaries. The male in the testes will be creating sperm, and that is the only place that you'll find gametes or 1N haploid cells in this individual. The egg and the sperm will then come together to fertilize a new individual. And this is those particular cells that are inside these adults that are creating the eggs and the sperms. Those are the germ cells. Germ cells, again, they are diploid. They are 2N cells that are going to go through meiosis to create the haploid gametes. So meiosis, finally, we are here. Meiosis is going to have 2 divisions. When we talked about mitosis only had one set of division, where it went through prophase to prophase to telophase, only once, and then cytokinesis was only happening once. Meiosis is going to be doing that twice. There will be 2 sets of these different phases, with 2 times the cytokinesis taking place. Germ cells are going to be going through interphase. Remember, interphase was the G1 phase, the S phase where the DNA was replicating, and then the G2 phase where all of the organelles and proteins were replicating and getting ready for cell division. Just like in mitosis, meiosis is going to have cells that are living in that interphase situation. And there is only one S phase, so the DNA will be replicating only once. So the DNA replicates, and then it's going to go through its 2 divisions as is. It's not going to replicate again. There will be 2 consecutive nuclear divisions. And so those divisions will be meiosis 1 and meiosis 2. And so the picture at the bottom, well, all on the right, is kind of showing this taking place. And so it's saying that, in meiosis 1, homologous chromosomes are going to separate. In meiosis 2, sister chromatids are going to separate. And so we'll look at this. Both pictures are showing the same thing, just some of them might be easier to see. So in the bottom picture, this germ cell, it has 2 pairs of homologous chromosomes, 2N, N equaling 2, so there are 4 chromosomes total. It has already gone through the replication, and you know this because these chromosomes have doubled, and so this is in prophase. It's ready to, the condensing of these chromosomes has taken place, and it's ready to go through meiosis 1. And so what will happen is these homologous chromosomes, and you can tell these are homologous because they're the same size, or at least this is what it's depicting. So you've got this small set of chromosome number 2, the large set of chromosomes number 1, and after meiosis 1, you're going to split the homologous chromosomes apart. So once that first splitting happens, you're going to have one big chromosome, one small. One big, one small. And you can see how the when this cell was created, the homologous chromosomes, one's coming from the mother, one's coming from the father, those are being split. Now you've got only the one from the father, only the one from the mother when we're talking about chromosome 1. When we're talking about chromosome 2, the small one, this one came from the mother, this one came from the father. And we'll learn that these splittings of which one is going to go to which cell will be random. And that is for the diversity. Then, when it talks about the meiosis 2, the sister chromatids are going to split. And so what's taking place here, this is very similar to mitosis, where you now have the chromosome, the duplicated chromosome, and it will split where half of this will be going to one, and the other sister chromosome, or the other half, will be going to the other. So theoretically, during meiosis 2, these could be identical to one another. Now, without knowing about the term crossing over, they would be identical to one another. But crossing over is a possibility of changing that, and we'll talk about exactly what crossing over is. So there's a lot packed into this one picture, and I don't expect you to all of a sudden know everything about meiosis, but this is a quick overview of what's going on. We will now talk about all the little details that gets into it. So again, DNA is not duplicating in between meiosis 1 and meiosis 2. DNA is replicating only once, and that is in the S phase before meiosis 1 or 2 starts. That is the only time DNA will be replicating, and then when these meiosis 1 and meiosis 2 takes place, there will not be another replication taking place. So the parent cell is a 2N cell, and at the end of meiosis 2, you will have 4 haploid cells. So one parent cell, one diploid parent cell will create 4 haploid daughter cells. And that is one major difference between meiosis and what we saw in mitosis, where mitosis was a 2N parent cell creating 2N daughter cells. And so, just like in mitosis, this picture kind of gives an overview of what's taking place, even more so than the last pictures that we saw, where we're seeing meiosis 1, and then we've got 2 cells after meiosis 1, and then they split again into meiosis 2, and you can see some crossing over taking place, which we'll talk about, and then the independent assortment taking place, which again we'll talk about. These are terms that are going to be important in the differences between some of these phases. But a big take-home message here is that the nucleus will be dividing twice. We will have 2 times the division of the cells. So meiosis 1. During meiosis 1, each chromosome will be paired up with its homologous chromosomes. So in a human cell, all 23 pairs will come together. The pair of homologous chromosomes will actually come together and kind of connect to one another to create a tetrad, to create a 2X shape. So they will actually be connecting to one another. This is important. The chromosomes will not be connecting to other chromosomes. You won't have chromosome 1 connecting to 22 or 5 connecting to 15. It's going to be homologous chromosomes 1 and 1 are going to come together. 2 and 2 will come together because they are important. They have the same genes on them as one another. And then when they come together in the beginning of meiosis 1, they will be the ones that will be separating from one another to go to the different cells, so that after meiosis 1, each daughter cell will have one of each of those chromosomes. They'll have one of chromosome 1, one of chromosome 2, one of chromosome 3, and so on. And so each daughter cell will have a set of 1 through 23. It's just a different one than the other cell. In chromosome 2, in meiosis 2, the two sister chromatids are going to be separating to each new daughter cell. And so this is what is taking place in mitosis, where the chromosome, you've got a duplicated chromosome that's in that X shape. And so after meiosis 2, that X shape will be torn apart, and you'll have the single sister chromatids going to opposite poles of the cell. So the stages of meiosis, and when certain questions are being asked about what phase of meiosis something particular happens, it is very important to tell me that it's prophase 1 or anaphase 2. If you just say the word metaphase, in this case, there are 2 metaphases. And so if something different is happening in each of those metaphases, the answer metaphase might not be enough. And a lot of the times it's not enough. For me, in our tests are multiple choice. And so it's not a huge deal because I'm giving you the choices, but you do need to know the difference between 1 and 2. When it comes to your lab practical, this is a big one. You need to know the differences between meiosis 1, meiosis 2, and all the different phases 1 and 2. And a lot of the times, some of these questions that are being asked, if you give me the wrong one, then that means that question is incorrect. So that being said, meiosis 1, we have, just like mitosis, prophase 1, metaphase 1, anaphase 1, telophase 1. We do not have a prometaphase 1, which would be found between pro and meta, and we do not have a prometaphase 2 that would be found here. But that is when knowing these different phases, if you remember all the phases of mitosis, it will be easy to remember the order of the phases in meiosis, in meiosis 1 and meiosis 2. But just remember, there is no prometaphase that we are discussing in meiosis. It could exist as a phase, it's just getting into the details that we're not. And then meiosis 2 has each of those phases. They're just happening in the second splitting of cells. So prophase 2, metaphase 2, anaphase 2, telophase 2. And there are some differences that are taking place between the 2 prophases, the 2 metaphases, anaphases, and telophases. So it's good to know all of these differences and similarities. So prophase 1, just like mitosis, we're going to go through each of these phases, and then we'll do an overview and kind of show the bigger picture. But now we're going to look at each phase and look at how the details that's taking place. So in prophase 1, just like prophase in mitosis, there will be some similarities here. The chromosomes are going to be condensing. During the interphase before meiosis takes place, everything's going to be leading up the same. And so the DNA will be in this chromatin form, this loose kind of stringy DNA. And so as we're approaching prophase, all of a sudden these sister chromatids are going to be condensing. One big difference between mitosis here, the homologous chromosomes are actually going to be coming together and joining one another. So pairs with homolog, the homologous pairs will be coming together and they will be connecting to one another to form a tetrid. And this connection is called a synapse. And the shape is called a tetrid. And so this picture up here is showing that these homologous pairs are coming together and forming this shape so that they're physically connected to one another. We have this possibility of crossing over. Crossing over is the switching of segments from one chromosome to another. There will be some pictures later showing more detail, but I kind of want to give you an idea of what's happening. Let's everybody cross your legs at the ankles. And so this is kind of similar to how these two chromosomes are going to be connecting to one another. Now, when your legs are crossed, think about your feet switching to the different legs while they're crossed at the ankle. When you then disconnect, you stop crossing your legs and you take them apart. Now all of a sudden your right foot is on your left leg and your left foot is on your right leg. Now, obviously this is impossible for your legs and your physiology, but that is what is taking place with these chromosomes. They are crossing tips and breaking the different tips off and switching to the next to the other chromosome, so that when they come apart, the different feet will be on the different leg, sort of situation. And this, we'll see, for the reason being, is for diversity. It will create the possibility of diversity down the line, making it much more diverse, possible diversity. So, in prophase 1, it is very important to know that crossing over is taking place here. We've got these tetrads forming, the possibility of crossing over. And so when I say possibility, we have 23 pairs of chromosomes that are pairing up, and not all of them will go through this crossing over. Some of them will. Sometimes more of them than others will happen. Where the crossing over takes place is quote-unquote random. There is some things guiding this along, but it is in a random type way that this is taking place. And so, this aside, we're now the next thing that's happening in prophase, and this is also what is happening in prophase of metaphase, of prophase of mitosis. The nuclear envelope is breaking down, and so we see, this is kind of starting to break open and dissipate a little bit. Spindles are forming. This is also happening in mitosis. And these spindles are going to be attaching to the kinetocores of the chromosomes. And so all of these things are taking place in prophase, and this is happening in prometaphase of mitosis. In metaphase 1, the spindles will be centering the tetrads. Again, the tetrads are those pair of homologous chromosomes that are attached, and they will be lining them up along the equator. Now, this is not the greatest of pictures because these should be attached to one another. Because this is a tetrad shape, they should be attached to one another still. So these homologous chromosomes are being lined up along the center. So for us, a human being, we would have 23 pairs of them lining up in the center. And they would be set up in a way so then when they break apart, one of these will be going in this direction, the other will be going in this direction. As you can see, this is the part that crossed over, and that's why the different colors are present on the same one. And that's probably why they drew this picture in a way, so you could see this crossing over. These will be in a shape, they'll be lined up in a randomly on which one is going in which direction. And so in this case, we've got the blue on top and the red on the bottom, but it easily could be flipped around with red on the top and blue on the bottom, just like over here. And so this randomization of, which of the homologous chromosomes are going to be facing one pole versus the other is going to be random. And when they're split up, that randomization is actually going to be creating diversity amongst the gametes. Anaphase 1, spindles will be tearing those tetrads apart. So each pair of homologous chromosomes will now be being broken and pulled apart, where one is going to one pole and one is going to the other pole. Sister chromatids are still attached to one another. So each of these is still in this X shape, this duplicate chromosome shape, where the sister chromatids are in the same, still attached. Now, as you can see, it's showing, there is some crossing over that had happened. In this case, this is a good time to look at this. The crossing over only took place on one of the sister chromatids. It did not take place on both. So when that tetrad is connected to one another, crossing over is not necessarily happening to both of the sister chromatids. It's only happening to the one. And this is what's going to create the diversity down the line when meiosis II is over. In telophase 1, the chromosomes will be arriving at the opposite poles. As you can see, the nucleus is starting to form around the chromosomes at each of those poles. It seems a little redundant that this is taking place because it's going to split later. But the splitting of meiosis I and meiosis II, sometimes there could be examples where there's going to be a time frame, a lag, if you will, between meiosis II taking place after meiosis I. So the nucleus will be forming around these chromosomes, which is happening here. The pinching, the cleavage is going to start, and we're going to have the cytoplasm starting to divide, where it's going to be going through cytokinesis to split into two cells. This is the first stage when they separate through cytokinesis. This will be the first time that we're going to have haploid cells. They are going to be 1N, each with one type of chromosome from parent cell. Now, you could be looking at it and being confused by that. So, this chromosome, yes, it's a duplicate because it's in that X shape, but this is chromosome 1. This over here is chromosome 2. When the cell was the parent cell and the germ cell, there was two of this. The homologous set was this one and this one. The homologous set of this one and this one. But we have taken those apart. We now only have chromosome 1, we only have one here, and chromosome 2, we only have one there. Same with this one. So this is the first time that the haploid cell is seen. So once that cell, and we didn't kind of get into the fact of the cytokinesis, but it kind of combined that together. The cytokinesis took place, and we have two daughter cells now that are approaching prophase 2. So after telophase 1, we now have 2 cells, and those 2 cells are going to be entering the prophase 2, or they're going to be entering meiosis 2. And prophase 2 is the beginning of this. And so in this phase, prophase 2, spindles will be attaching to the chromatids. The nucleus, we just saw it starting to form again, and now it's going to degrade and dissipate and kind of expose the DNA back to the cytoplasm again. The microtubules that are attached to the kintochores of these chromosomes, they're going to line them up in metaphase 2. And so this might look familiar. This is kind of what mitosis looks like, the metaphase of mitosis, where the chromosomes are lining up in single file. This is different than meiosis 1, the metaphase 1 in meiosis, because that is when we saw the tetrads lining up, so double file. But meiosis 2, we have the chromosomes lining up in single file. Now you can see the differences here. Each cell it's happening in, and you can see that each of these cells are slightly different, the colors and everything that's going on. So they're already diverse from one another, and you can visually see that. And we're only talking about 2 chromosomes. If you had an organism like humans where you had 26 chromosomes, just think of all the diversity that you could be seeing. Anaphase 2, the spindles are going to be, the microtubules will be pulling those sister chromatids apart. And then the chromosomes will be moving to opposite ends, the opposite poles of the cell. And even though this is the same chromosome that's being pulled to one end, you still see that there is some differences that are going on. That chromosome has some diversity amongst it because of the crossing over that took place. But the sister chromatids here, take-home message for this phase, the sister chromatids are being broken apart and separated to their poles. Telophase 2, the chromosomes have arrived at their poles. The nuclear envelope is going to be reforming, and cytokinesis will be taking a place again. So the cleavage will start and pinch, and then cytokinesis will follow, and you will have 4 haploid cells as a result. So each of the cells that arrived, that started in meiosis 1, will be going through this. Each of them will be separating into 2, and that's how we now have 4 cells, 4 haploid cells. And so you can see that the DNA chromosomes in each of these and how each one of these is slightly different than the other. So we now have 4 different haploid cells created from one germ cell. Now this is a more complicating overview, but it does, for some of you this might be helpful, kind of showing you all the different phases, and so looking over that might be helpful for you. Basically, you know, the G1, so this is kind of talking about meiosis altogether. Interphase G1, S, G2 is not present. It's kind of just a little snippet right here. The prophase 1, metaphase, anaphase, telophase 1, it's splitting, and then cytokinesis, and then each of these are entering into meiosis 2, and you see them going through prophase 2, metaphase 2, anaphase 2, telophase 2. They're being pinched through cleavage, and then cytokinesis, and now you had the 4 cells, the 4 haploid cells. So 2 important events took place in meiosis, and they took place leading to genetic variability in the daughter cells. And as you might have guessed it, they are crossing over which took place in prophase 1. Independent assortment, this random alignment of homologous chromosomes that took place in metaphase 1. You should know a little bit about what had happened because we talked about those, but we're going to get into each of them a little bit more in depth so that you understand. But make sure that you know crossing over took place in prophase 1. That is the only time that crossing over is going to take place. Independent assortment is taking place in metaphase 1. It is the only time that this is going to take place. You will see questions like this on the test, on the lab practical, and you'll have to remember that those are taking place at these times. Remembering that they took place in meiosis 1. Both of them are taking place in meiosis 1. And then if you understand what crossing over is in independent assortment, you should know what phase that's taking place in. So this slide is devoted to crossing over and what exactly is taking place. My great metaphor of crossing your legs and switching feet, if that wasn't helpful, this is kind of showing a little more detail of what's happening. So the tetrad is forming, so these two homologous chromosomes. We're not dealing with blue and pink now, we're dealing with red and green, and it doesn't really matter what parent they come from. So these two are homologous chromosomes. They could be any chromosome 1 through 23 if we're talking about a female. And so they come together and they're going to be connecting to one another. The non-sister chromatids will cross and break at the same places. And so when we say non-sister chromatids, these two red, the one at the top, the one at the bottom, those are sister chromatids. They are connected to one another. They're identical copies of one another. One was created during DNA replication. They were created. They are identical copy to one another. This chromosome over here, same thing. The top and the bottom are sister chromatids to one another. So when we say non-sister chromatids, we mean this one, this green one, and this red one. And the crossing over here is taking place. As you can see, the red and the green non-sister chromatids have crossed over. They've crossed their legs. There will be an exchange of corresponding segments, genes, at a particular breaking point. And so this switching here that's taking place, it could be here, it could be here. It doesn't have to be at this exact place. It could be lots of different places along this DNA strand. But usually what's going to take place is it's going to break not in the middle of a gene, it's going to break in an area that is between genes, so that it will not be messing up a particular trait of that organism, a particular protein that will be created, because that could cause some damage down the line. So even though it's random along the DNA strand where it's going to break, it's not random exactly where it's going to break. There are particular spots that it could break up and down the line. The resulting chromosomes now contain genetic information of both the mother and father. So this chromosome, let's say, came from your father, but this end of that chromosome did not. And so, so it's now a mixture of the two. And that is crossing over. So some more depictions of crossing over in case it wasn't grasped. So crossing over is taking place in meiosis prophase 1. During crossing over, two homologous chromosomes pair up and exchange pieces, scrambling the genetic material. And so we can see, this blue and this pink, and up and down the line you see the different traits on the chromosomes. So this chromosome, is identical to one another. These sister chromatids are identical. These sister chromatids are identical. But the non-sister chromatids are not identical. Yes, they have A through F, but this one is all capitals. This one is all lowercase. So that means they're different alleles from one another. During crossing over, these two non-sister chromatids are going to cross their ends and switch. And so you see that the crossing over, we're not seeing the exact placement here, but it's exchanging this portion for this portion. And after meiosis 1, after prophase 1, and after crossing over, you see this portion where the alleles A and B are now on this end, and big A and big B are now on this end. This is all for diversity of our gametes, so that there will be lots of differences amongst our gametes. So random alignment, random assortment, random alignment, these are kind of synonyms to one another for this phase, for this event that's taking place during metaphase 1. Metaphase 1 of, obviously, meiosis 1. And so this is the random placement of our homologous chromosomes, where the microtubules are going to attach to the chromosomes, and it's going to kind of randomly put them in a way that, some of the blues are going to go up to the one cell, and then some of the pinks are going to go up to that cell. So not all of the chromosomes from your father are going to be going to one cell, or not all of the ones from your mother are going to be going to one cell. It will be a mixture of the two parents. And so this is a good depiction of what's going on here. So independent assortment, random assortment is occurring in meiosis metaphase 1. In independent assortment, chromosome pairs align randomly, scrambling the combination of chromosomes for each gamete. And so if we look at this example, this does have all of the blues at the top and all of the pinks at the bottom. Now this is statistically, that can happen, especially if you only have if you're only dealing with 3 homologous pairs. In a human being, we're dealing with 23 homologous pairs. And so for this to happen, it's very low statistically. It is possible, but it's very low. And so I go back to the picture. So this version, all the blues are at the top, all the pinks are at the bottom. When we look here, we can see there are 2 blues and 1 pink at the top, 2 pinks and 1 blue. So this is one version of what can happen. This is one way that they can align differently, and this is another way that they could align differently. And so these are all the different ways that they could line up when you're dealing with 3 homologous pairs. And then when they go through meiosis, and then meiosis 2, you're going to have these versions. And so independent assortment produces many chromosome combinations. A germ cell with 3 pairs of chromosomes, 3 homologous pairs of chromosomes, will have 4 possible arrangements during meiosis 1. And so that's what we were looking at here. 1, 2, 3, 4. These are the 4 different ways that they can line up. And then they're going to yield 8 possible gametes. And so if you're dealing with 3 homologous pairs of chromosomes, you can have these 8 different gametes. Well, in a human being, we have 23 sets of chromosomes. And so if you take this math equation, there are 2 of each chromosome, so you would take 2 to the 23rd. that equals 8 million, almost 8,400,000 different gametes. That is how many different gametes one individual can make. And that is just looking at this independent assortment. Theoretically, an individual can have 8,388,000 different offspring, not even offspring, different gametes, different sperms, different eggs. And that's just independent assortment. When you throw in crossing over, now you have the possibility of mixing up these different pieces of chromosomes. And so I say the possibilities are endless, that it's infinite amount of differences. I don't know if that's 100% true, but you can see how extraordinarily large that number can get when you're including independent assortment and crossing over. So those gametes that are created, those eggs and those sperms, they're going to come together to fertilize one another and create a new organism. They will restore this diploid chromosome number, where one gamete is 1N, the other gamete is 1N. You put them together, you're now going to have a diploid 2N cell. The male and female gametes, the sperm and the eggs, will unite, creating a zygote. Each cell contains a pair of chromosomes. Which two gametes are going to unite? So when we looked at all of the different possibilities of all these different gametes that are forming through random assortment, through crossing over, is any of them more special than the other? Is it going to be able to, is that special sperm going to be able to get to the egg any better than any of the others? I'm going to say no, that they are going to unite randomly. There are some cases, I won't get too much into it, but there are some slight differences between male sperm and female sperm. So when I say male and female, some of the sperm will have an X chromosome, some of them will have a Y chromosome. It's a very slight difference, but it is a difference in weight. And so some of those sperms are able to swim faster for a longer amount of time because they're not having that weight. And so there are some differences. But for the most part, when we're talking about the DNA, these gametes will be uniting randomly. And this will lead to genetic variation. The randomness of this is going to lead into genetic variation. Just one more step in how genetic variability can take place. Now, we were talking about one egg being fertilized and creating one zygote. And as we know, triplets, twins, triplets, quadruplets, these are all possibilities. And so we'll talk about the different versions that can take place. So monozygotic twins are genetically identical to one another. So how is that taking place? Sometimes during early development, when an embryo is being created, it will split into two. Each embryo then develops independently. So here we've got the sperm fertilizing the egg, creating this zygote. The zygote then becomes 2 cells, 4 cells, 8 cells, 16, 32, 64. Well, at some point, that 64 cells starts to break apart and become 2 groups of cells. So each group is going to be identical to one another because it has the same DNA in them as the zygote did. And so when this happens early on, you now have 2 embryos that are forming. Each of those embryos are going to be identical to one another, and you will have identical twins. They will share the same DNA as one another. They'll both be the same sex. They'll both be identical to one another. Now, if you get into higher biology classes, you will learn that there are some differences between the two, especially in their immune system and things like that, genetically. But for the most part of what we're learning, they are identical in all ways. Not their fingerprints and things like that, because those form in the womb of the mother. Dizygotic twins, they're going to have unique DNA. So when you see twins being a male and a female being born together, this is an example of dizygotic twins. They do not have to be two different sexes, but they will not be identical. And so this is when, so this is called fraternal twins. The monozygotic is also called maternal twins. But dizygotic and monozygotic are more biological phrases here, terms. Basically, there could be 2 eggs released at one time inside the womb of a female. And when 2 eggs are present, 2 sperms come in and they fertilize each of the eggs. A totally different sperm will be fertilizing this egg from this sperm, which is fertilizing this egg. And so it's basically 2 children are being born, or 2 zygotes are being created at one time. Both zygotes are completely unique to one another because they're different eggs and different sperms that have been fertilized. Those embryos will be unique to one another, and then the children that are born will be unique to one another. So 2 sperm fertilizes 2 different eggs, and the offspring might look similar to one another, but they're going to be very different individuals. So a synopsis here of mitosis and meiosis. And so this is a good way of kind of studying, making sure that you know the differences and similarities between the two, making sure that you know some of these phrases and what's going on here. And so mitosis, you've got the interphase, which is the same between mitosis and meiosis, the G phase, S phase, or G1, S phase, G2. And then in prophase, we're going to have, in meiosis, the tetrads are going to be forming. Crossing over is going to be taking place. That will not be taking place in mitosis. In metaphase of mitosis, you will have the chromosomes lining up single file. In metaphase 1 of meiosis, they will be lining up double file. They are homologous chromosomes. These tetrads will be lining up along the equator. And this is where you're going to have random assortment taking place. In anaphase of mitosis, you will have these sister chromatids being pulled apart. And then you will have the daughter cells that are identical to the parent cell. And this is anaphase and telophase combo and cytokinesis. Whereas anaphase 1, telophase 1, you now have the beginning of this haploid cell, which is, they are not unique to one another. They are not identical to one another. They are unique. And you can see that by the DNA inside the cells, inside the nucleus. And then it doesn't end here with meiosis. It then has to go through meiosis 2. And then you'll see in metaphase 2 of meiosis, this is when they will be lining up in single file, very similar to this. And then at the end of meiosis 2, you will have 4 haploid cells, 4 haploid daughter cells, and if crossing over took place, then they will all be unique to one another. They will all be slightly different, if not majorly different from one another. Mitosis, the functions here are asexual reproduction, growth, repair of multicellular organisms. With meiosis, it's sexual reproduction. Mitosis, we have, this is occurring in somatic cells, 2N. The parents are 2N, and the offspring is, the parent cell is 2N, and the daughter cells are 2N. For meiosis, it's going to occur in the germ cell. The germ cell is 2N. Do not forget that the cell that starts meiosis is going to be 2N. This 2N parent cell will be creating the 4 unique haploid cells, 4 1N cells. So, just like in DNA replication, just like in mitosis, just like in all these different processes, errors can take place. And when these cells are dividing, errors can take place in the number of chromosomes that are going to be going into these different cells and how they're going to be splitting. It is not as prevalent as you might think, but these errors do take place. Gametes can end up with the wrong number of chromosomes. The consequences can range from a minor issue to very lethal. If that particular gamete, that particular sperm, with the wrong number of chromosomes fertilizes an egg, you can have consequences here of minor to lethal. Options here: polyploidy. These individuals have 3 or more of each type of chromosome. So when we talk about where a cell is haploid, which is 2N, a diploid cell is 2N, a haploid cell is 1N. If for whatever reason in the splitting of a gamete, the gamete ends up being 2N, it took all of the DNA with it, and then it fertilizes an egg that's 1N. So you have a 2N plus 1N is going to have the offspring, the zygote of a 3N individual. In humans, this is lethal, and this zygote will not survive. But in other organisms, plants, they're able to thrive this way. It's a unique situation. They can only then be able to mate with other 3N individuals and things like that. But in other organisms, this can happen and still thrive. So again, that polyploidy is the 3N, 4N, the number of the entire set of chromosomes. It has not only 2 of each homologous chromosome. This has 3 of each homologous chromosome. This has 4 of each homologous chromosome. Aneuploidy, aneuploids. These are individuals that have 1 extra or 1 less chromosome. So the offspring, the zygote will be 2N, but 2N plus 1 or 2N minus 1. So it'll have, in this case, we'll have 2N, so it has 23 pairs of chromosomes, but then it has an extra chromosome number 7. Or in this case, there are 23 pairs of chromosomes, well, 22 pairs of chromosomes, but then chromosome number 7 only has 1. It doesn't have the second chromosome to go with it. And this is due to non-disjunction. So the term non-disjunction is what is causing this to happen, aneuploids to happen. And there is some pictures in the next slide, or on the next slide. So non-disjunction, failure of chromosomes to separate correctly during meiosis. If there is one too few, then that is monosomatic. So this is the case where 22 pairs of homologous chromosomes, but one of them only has one. And so in most cases, that individual will not survive. Trisomatic is when this is this 2n plus 1. If one too many chromosomes are there. There are a few cases of survival, and usually there are some kind of handicaps involved with the individual. Not everything is normal case. When I say normal, there are some issues taking place here. And so non-disjunction, this picture is kind of showing during meiosis 1, it split. And so this is talking about the X chromosome and the Y chromosome. We could be discussing chromosome 7 on both of these or whatever. It's just giving an example. And so after meiosis 1, this homologous chromosome went here. This one went here. That's normal. Then in meiosis 2, this one split normally. This one did not. Instead of the sister chromatids being split evenly amongst these, for whatever reason, both of these went to this one and none went to this one. So if this were the sperm or the egg, and then all of these were to fertilize, if this was sperm, and you would see all of these fertilizing an egg. These two would fertilize the egg, and potentially you'd have a normal individual, a normal genetic situation. If this sperm were to fertilize an egg, you would have that trisomatic situation where one extra chromosome is there because this is going to match up with another from the egg, but then you've got this one by itself that's that trisomatic. If this fertilized the egg, then you would have one less fertilizing the egg. So the egg would have that chromosome, but you would not have a homologous chromosome matching up to it. There are lots of different examples of different things, different situations that arise from these trisomatic situations. Down syndrome is one example of a trisomatic event that's taking place. This is an autosomal chromosome number change. So the trisomy of chromosome number 21. So that means that there is one extra chromosome 21. Individuals that are born this way usually have mental impairment and a variety of additional defects. Can be defected before birth. And here is an example of the chromosomes. As you can see, each of them are paired. But then when you get to 21, you see that there are 3. So this karyotype is showing that there are 3 chromosome 21s. And that is what is causing this situation to take place. Another case is Klinefelter syndrome. And so this is a trisomy non-disjunction of the X chromosome. And so this is the sex chromosome where there are 2 X's going to the new organism. So the most likely, I believe it I guess it could be the egg or the sperm, where if it was the egg that had the non-disjunction, both X's would be coming from the egg. If the sperm had the disjunction, it would be the Y and the X coming from the sperm. And so either way, the zygote that is formed would have an XXY situation. So instead of XX or XY, it has this extra sex chromosome. The phenotypes, the individuals tend to be very tall. They will be sterile males. And a treatment for them is treating them with testosterone to kind of get them some of their effects lessened. And here are some symptoms of this situation. And you can see in the karyotype, you've got the two X's and then the Y. So before I had said that monosomatic examples would be fatal. There are some examples where there can be some survival. This situation of a monosomatic situation is Turner syndrome. And this is where the individual is only an X. They did not receive the second X chromosome or a Y. So they just have one X chromosome. 98% of these cases spontaneously are aborted. They naturally miscarry. They will not survive. Survivors tend to be short individuals and infertile females. They can be treated with hormones or surgery to lessen the symptoms. Other chromosomal abnormalities can take place. There can be, a lot of the times, these cases happen from crossing over, where crossing over has some problems. You can have the deletion of a part of a chromosome. So maybe during crossing over, one chromosome gets both of those ends, and then the other one doesn't get one of the pieces at all. And so you can have one chromosome having that deletion, and then the other one having duplication. So the copies of a part of a gene are more than one. And so these are kind of, when this happens to one, it most likely this is happening to the other during crossing over. You can have inversion, where that portion of DNA did not connect correctly. It was flipped, and it's just not connected correctly. There's no good picture down here. And then translocation, the chromosomes are exchanging at different parts. So maybe instead of connecting at one end, it's going to the other end of the DNA and connecting there. And so these pictures kind of show where after the crossing over, you've got different size DNAs and different things that are taking place. This is a case of a deletion example, the Creducat, and so this is the deletion on chromosome 5. The larynx does not form properly, respiratory problems, behavioral problems, and mental impairments. And so if you think about this, there are certain portions of certain pieces of DNA that are, happening more often than others. And so it just kind of shows that the randomness, quote unquote randomness, of where these chromosomes are switching, the crossing over is taking place, might not be exactly random. There are particular spots that it could be taking place more than others. Meiosis produces human gametes, as we've been talking about this whole time. Germ cells located in the testes give rise to sperm cells. Germ cells located in the ovaries give rise to egg cells in females. And so spermatogenesis is the production of sperm cells from the germ cell. And so this is, as you might assume, this is exactly what we've been talking about. where my mitosis creates these germ cells, and then the germ cells go through meiosis 1, meiosis 2. You get the 4 haploid cells from the 1 germ cell. And so this is spermatogenesis, and this is what you would expect, what we have learned. Oogenesis is actually slightly different than what we've been learning. And so this 1 germ cell is going to produce 1 egg cell from each germ cell. So this germ cell is really only going to be creating 1. And so let's kind of go through. So through meiosis 1, we'll be creating a cell during the cytokinesis, 1 cell, and then the other one is considered a polar body. And so even though that these are the same size, this polar body is going to be a lot smaller, and then this cell will be a lot larger. And then through meiosis 2, this polar body will then be going through meiosis 2 and creating 2 smaller polar bodies. This cell will be also splitting into 2, creating 1 polar body and then 1 larger egg. And so the reason for oogenesis creating 1 egg, which in reality it is creating 4 cells, it's just creating 1 egg. The 1 egg is going to be much larger than these polar bodies. It's going to have much more cytoplasm, much more of the organelles, and so it is getting ready to be a healthier beginning of this zygote. And so when the sperm comes in and fertilizes this egg is ready to go. So yes, it is producing 4 haploid cells. Even though these are called polar bodies, they're still cells. They're just not going to be functioning egg cells. They will be reabsorbed, and this 1 egg will be the one that's created to be released for fertilization. Oogenesis.